Hello, everybody, and welcome to this session. My name is Natalia Jimenez, and I'm the director of the HPC AI and Quantum Life Sciences Center of Excellence of Atos. I'm delighted to be here today to show you some of the use cases of advanced technologies such as high performance computing and quantum in the life sciences industry. But before doing that, I would like to frame the whole Atos value proposition, which focuses in helping researchers to overcome the challenges that arise in the life sciences projects. I'm illustrating this with this simplistic workflow where we have a sample that we put in a high throughput device to analyze it. The output of this device is raw data, and we need to transform it into actionable insights. To do that, we may use a combination of different technologies. Atos works with researchers to find out what is the right combination of those technologies. Today, we are going to focus primarily in high performance computing to accelerate the processing of the raw data that we get from these high throughput devices. But to make this presentation more aligned with the current situation, I decided to organize it in three main steps in the fight against SARS-CoV-2, the pathogen that is putting our world inside out. Let's begin with the structural biology techniques that enable researchers to make visible or invisible enemy. This technique is called 3D electron microscopy. Well, as you can see here, we are going to focus in the spike of a virus, which is called protein S. And this is the responsible to bind with the cell receptor initiating the infection. What we are describing here is a typical workflow in 3D electron microscopy. First, you need to freeze very rapidly your sample to prevent the formation of crystals. Then you take the pictures with the electron microscope. You can take hundreds of pictures in one go. This is how your specimen looks like in one of these pictures. Well, it seems that it's not very promising, isn't it? So how do you get from this here to this kind of representation? Is a very complex processing workflow where you need high performance computing. In this case, the author picked up more than 630,000 spike proteins, and then they need to do the alignment and all the different steps of a process to finally align and add them together, the different views of this protein, to combine them to generate a 3D model. That is what you can see here. And this has been published in Science, and this is the work of a laboratory in Texas. Um, so that's here we can leverage high performance computing, AI, and big data analytics to help researchers to accelerate these processes. Um, Here you have well, it's the same specimen, is the spike of a virus, but here this is a refinement. The author took the high resolution model published in Science to better refine, to reach to a higher resolution and to observe this flexibility of the spike of a virus. This is a preprint that will be published very soon. Um, and indeed, those authors are part of a biocomputing unit of a National Center for Biotechnology, which belongs to the Spanish Research Council. A part of, well, being focusing in COVID-19 now, they lead a project to gather all the tools and software to process to process the output of a electron microscope. And this is part of a project, a European project called Instruct, and they are the image processing partner of this project. Well, as I said at the beginning, the spike protein or protein S binds to the cell receptor, allowing the virus to enter into the cell where it can replicate using the cell machinery. Proteins like these are called target molecules because they are used as targets to develop drugs. As you can imagine, knowing the 3D model of this is key in the fight against any disease. If we prevent this binding to happen, the infection won't never take place. This links with the following section, which is called Getting Ready for the Fight. So once researchers have identified a possible target, in this case the spike of a virus, they can test a library. I need to come back to this one. They need to test a library of compounds against it. In some case, in some cases, the library can be as large as 10 over 60 different drug-like compounds. And this process is called drug screening, and this is the first step towards finding a treatment. The following step is the preclinical testing, when are taking the most promising lead compounds and you are testing them normally in animals. Then you move to humans for the clinical trials, and then you must follow the FDA or the EMA approval. So, molecular dynamics and molecular docking are two main methodologies that researchers use in the drug screening process. In the first, you simulate the movement of the atoms of a macromolecule, in this case the protease of a virus, which is responsible of its replication once the virus is inside the cell. In this video, you can see how these kinds of simulations can reveal new binding sites. As you can see here, eventually we will see a pocket in this structure, which is exposed. And this obviously this would be a target for developing new drugs. With molecular docking, you simulate a drug binding to that pocket. All these simulations are, as you can imagine, computing. You can see here this yellow structure. This is the drug molecule, and you are trying to fit or to dock it in different positions inside that pocket. So, as I said, these simulations are compute demanding. Continuing with the clinical trials, this part or contribution in this area is this proof of concept where we tested the validity of quantum simulator to identify comorbidities in clinical trials. We did this in collaboration with Bayer and with Aachen University in Germany. But imagine we can design drugs to ensure they end up in the right place and attach to the right proteins. We are all unique, and the efficacy of different drug delivery mechanisms varies by person. Imagine we can create a virtual human, a digital representation of you using computational modeling. On your virtual avatar, we can explore different delivery mechanisms to find the most effective. This is not science fiction, but the Conviomed project, an European funded project led by University College of London, and whose main goal is to create the virtual human by compute modeling. The third point I'm going to speak are the immunological studies, which is what most countries are doing now to check what is the percentage of a population already immunized. But there are many other things that can be studied if we add genomics to this picture. If we sequence the whole genome of people infected and we correlate this with electronic medical records, we could advance in our knowledge of the disease. And we could be potentially segmenting patients in different cohorts and answering questions such as why some people are asymptomatic, what variants will determine the severity of the disease. If we have a look at the genomic pipeline, and this is again a very simplistic representation, we will see a first part of a pipeline which is compute demanding and requires high performance computing to be accelerated. This is the part in which we do the genome alignment, where we get the patient genome and we have a reference genome, and we try to map all those different pieces that are the reads, and we have millions of reads that are at the end of the day pieces of DNA against this reference genome. But then there is also the data analytics or data demanding part, which is this one when we do the variant calling or the variant annotation, and finally we generate the report to be used by the medical doctors to diagnose or to prescribe a personalized treatment. Atos has been partnering for more than a decade now with the National Center for Genomic Analysis. This is probably the second largest center in Europe in terms of sequencing capabilities, right after the Sanger Institute in the UK. SINAC delivers services to pharma companies and hospitals with human sequencing. In this center homes 12 latest generation DNA sequencers that produce the equivalent to 8 whole human genomes per day. Apart from genomics, they have been involved in many interesting projects, such as, for example, the sequencing of the albino gorilla or Iberian lynx, identifying in the first case what are the genetic variants responsible of the albinism, and for the second, helping in its preservation. The Pilbright Institute, funded by the Biotechnology and Biological Sciences Research Council, focuses in the prevention and control of viral diseases in livestock and poultry. One important thing to highlight here, very related with the current crisis, is that Pilbright also studies those viruses that are jumping from animals to humans, what is called zoonosis. Genomics is a very important part of a picture, and in this case, heterogeneous supercomputer allows researchers to assemble genomes quickly and study the interactions between virus and host to understand how the virus evolve and how a model how they spread between individuals and farms. Atos also works very closely with hospitals and healthcare systems to facilitate the implementation of precision medicine. Our role is to enable the transformation of all the big data generated in the healthcare settings into meaningful insight by deploying our technologies. By doing that, we can turn the blood and fresh human being into its digital avatar. So here what we can see is, well, the normal human generating data by different technologies, could be omics technologies, and genomics is only one of them, could be pathology data, sensor, wearable devices, electronic medical records, or different modalities of medical imaging. And here, well, what we do is to enable all those data to be transformed into actionable insights. And to do that, we need the help of high performance computing, quantum learning machine, big data analytics, edge computing, to name only a few. And by doing that, we are facilitating the implementation of precision medicine, which is predictive, preventative, personalized, and participatory. All these case studies I've presented have been possible thanks to our capabilities in high-performance computing. Indeed, Atos is the top one in Europe, India, and South America, with more than 400 customers and more than 700 experts worldwide. Moreover, our quantum simulation program is the #1 worldwide. With all these capabilities in place, we've decided to create the HPC AI and Quantum Life Sciences Center of Excellence, which is a global initiative to bring technologies closer to research and clinical organizations, as well as pharma and biotech companies, to explore and test novel use cases. Located in Cambridge at the Biodata Innovation Centre at the Wellcome Trust Genome Campus, this COE will complement the existing campus compute facilities with Atos technologists collaborating directly with biodata research scientists to help boost campus innovation. In this center, we are exploring technologies such as HPC, AI, Edge, and Quantum in collaboration with researchers in different areas of life sciences. I encourage you to watch this video that explains more about the COE. Currently, because of a current situation, we are operating virtually, but please feel free, don't hesitate to get in touch with us to discuss on how to boost your projects. Thank you very much for your attention.